So Katja is here with us, and Katja uses neuroimaging to understand the evolution and development of the brain. And she is also strongly involved in open, reproducible science and interdisciplinary research. I'll be sharing some of her interesting, work in the chat so that you all can have a look into it. And it's up to Katja now. Please, share the screen and start with the talk.
Thank you very much for the kind introduction and for having me. Can you see my screen? Perfect. I would like to tell you something about the variability and stability of folding patterns across cortical development and evolution. And I'm working with Roberto Toro in his research group in Paris. And our latest research I would like to present that now. The cortical folding pattern is a characteristic of large mammalian brains, and here are some examples. This is a ferret that you can see to the left in the upper row, and then a sloth bear, and in the downer row there are some primates, the orangutan at the right, and then tufted capuchin, bottom left second image. And we observe that folding patterns are strongly conserved across individuals of one species, and their geometry corresponds with the cytoarchitectonic, connective, and functional organization of the brain. And when we look across species, both the geometric and the organizational complexity vary together. And we believe that this relationship may hold interesting information about the origin and shape of our cognition. Now, how does this organization develop and evolve? It's commonly thought that this organization is encoded genetically. As a side effect almost, folding patterns would also be genetically encoded. However,
Katja, just 1 second. Your other slides are not visible. Is this the only screen, or you have changed the slide because it's not visible to us? We can just see the first slide. Is that it? That's it?
No.
Yeah, now we can. Now we can. We could not see in between. So yeah.
Okay. Should I go back? So like this, you can see my screen.
Yes.
Okay. I was just showing that cortical folding is a characteristic across large mammalian brains. And these are some examples. And as you couldn't see them, I show them again. Here's a ferret brain and a leopard, a sloth bear, a black rhinoceros. And then down here, a primate brain with a gorilla to the right and a great-cheeked manga bay, a tufted capuchin, and a baboon. And what we observe is that the folding patterns correspond with the underlying cytoarchitectonic, functional, and connective organization of the brain. And when we look across species, both the geometric and organizational complexity vary together. And we believe that this relationship could hold interesting information about the organization of our cognition. And how does this organization develop and evolve is the question that we would like to answer. And it's commonly thought that the organization is encoded genetically. And almost as a side effect, you would have folding patterns genetically encoded as well. And there is, however, supporting evidence for the idea that folding may be purely mechanical, and raising the possibility that mechanics could play an important role in brain organization and development. And they don't play if it's not. Do you see the slides if it's now on show? Okay. What you can see here is interesting. It's simulations, both sides. And the left side shows a simulation that starts from an ellipsoidal shape. And it's just implemented homogeneous growth. So with homogeneous growth based on this ellipsoid, you arrive at having folding. Very interesting, and the folds also resemble to what we observe in cortical folding. And on the right side, we changed the geometry. It's still a simulation, but it starts from a smooth ferret brain at birth, where it's still lissencephalic. And just based on homogeneous growth, you can have folding and folding pattern if you give an initial geometry. And that's very interesting. So we aim at, I can't go to the next slide. We would like to better understand the role that mechanical morphogenetic forces play on the brain organization.
Try escape, maybe.
Okay, now it worked. Now you can see the slides again. I'm so sorry. Yeah, so we would like to better understand the role of mechanical morphogenetic forces on the brain organization. We studied an open data set of 65 different MRIs, individual MRIs across 34 different primate species. And we segmented the PL surface and did the 3D surface reconstructions. And also in a first analysis, we looked at the folding, the cortical folding. So how we analyzed cortical folding is we had a measure for the gyrification index where you have the folded brain and can measure the surface. And if you make a smooth envelope of the same volume as this folded brain, that's the ratio then you take for deriving the gyrification index. Then we measured the folding length, which is when you calculate the mean curvature and you cut off at 0 point and divide it by 2, like the dashed line that you can see here in the image. And a measure for folding wavelength. So where you start at the mid of the gyral wall and go down to the sulcal fundus, go up to the gyral hill and down to the midline again. So that's the folding wavelength we were using. And then we use phylogenetic trees built from genetic data to fit different evolutionary models. And I'm going to show you 2 of them if they play. This is the Brownian motion model where phenotypes are assumed to vary randomly along the phylogenetic tree. And if you have a speciation event, then they start varying independently, so they can diverge. And the more the speciation event is in the past, the more different the phenotypes are assumed to be. And this is the Ornstein-Uhlenbeck model, where phenotypes also vary randomly along the tree, but they have an adaptive, advantageous threshold that they all thrive towards too. And so we tested different models, and the one that fit our data best was the Brownian motion model. And interestingly, what we observed in our analyses, that you have some, like looking at brain volume, for example, you have some branches, for example, the one leading up to humans, where there's only a like increasing in brain volume. Then you have other branches that would, for example, lead up to the very smooth brains, like the
Katya, we cannot see anything. We are just seeing a black screen. Yeah.
So here you see the branch leading up to humans, where there's only an increase in volume. And you see other branches, for example, leading to the gray mouse femur or the galago, which are very smooth and little brains, where there's only a decrease in cortical folding starting from the common ancestor. And then you have branches where you can see like it's going up and down and up and down. For example, the one leading to the tufted capuchin here. And I'm pointing this one out because we're going to have a closer look at that one later. And the same, just differently, this is a phenogram showing the same. If you have the tufted capuchin here, you see that the line actually for the tufted capuchin ends up here, very close to, and we're going to look at that later, to the crab-eating macaque. So what we estimated based on that model, the common ancestor is supposed to have a brain that's very close to the now living II, so in terms of folding and brain volume. And what was interesting, what we observed is folding wavelength. Across all these species, when you have a 20-fold difference in brain volume, you had a very conserved folding wavelength of about a bit more than 1 centimeter, starting from the vervet monkey all the way like through all these species to the humans, with a very conserved folding wavelength. And this agrees with the mechanical folding hypothesis, where fold width is determined by cortical thickness, and cortical thickness varies very little across primates. And so we developed, this is the first approach that we took towards understanding the mechanical forces. And the second one is to develop a method to quantify folding pattern similarity. So we start from the mesh that we reconstructed. We compute the mean curvature and cut off everything that has negative curvature so that we are left over with a gyral skeleton. And then we shrink that into this wireframe that we can then represent as a graph, an adjacency matrix of the edges representing gyri and the nodes where the gyri would join each other. And the sulci are the holes that you see in these graphs. And here are different fold graphs across the different primate species. And we then use the graph edit distance algorithm to quantify the difference between these folding graphs. And I'm going to focus, interestingly, like we observed that the folding pattern similarity seems to be more strongly driven by brain volume than by phylogenetic relationship. And that's what we see here in these plots. So the tufted capuchin is an interesting example because its direct ancestors were supposedly having a very little and smooth brain, although then the capuchin derived a very large and folded brain. So this is, for example, very close in phylogenetic relationship, the Duruculi and capuchin. But if you look at the graph, you need to make more edits to derive a Duruculi folding pattern into a capuchin folding pattern. And on the contrary, when you look at the crab-eating macaque and the capuchin, there's very little edits that you need to make to the graph between the two species, although they have their common ancestor 47 million years ago and are phylogenetically less close to each other than the Duruculi and the capuchin. And this is interesting. So we propose to explain that folding patterns are regulated by geometry, and given enough growth, they will develop in the same way. And this is some sort of like mechanical canalization where the same structure develops despite a variation in genetic factors. And on the contrary, if folding patterns were encoded by differential gene expression, that would mean that exactly the same non-trivial folding pattern emerged in parallel in two different phylogenetic branches, and we consider that rather unlikely. And what's here's just like to be more clear where these species are in the phylogenetic tree, here and here. So their common ancestor very long, 47 million years ago. And while the crab-eating macaque, in terms of gyrification, was getting more and more and more, and then a little bit less. And the tufted capuchin had here this massive increase. And it's not an exception. So when we look across the entire tree, we see that the cebidae are colored in yellow. So that's the same branch as the tufted capuchin. And they are very tiny brains and little folded. On the contrary, the tufted capuchin is 47 million years from this one and from this one phylogenetically. And they are here in these different branches. So it's not an exception, it's different. This is the great-cheeked mangabe. We have the macaque, we have the white-faced sapajou, we have the tufted capuchin, king colobus. So that's something we observe not only as an outlier. And then another approach that we took towards this question was to reconstruct an evolutionary trajectory for cortical expansion. And so we did an alignment of these species based on volume, disregarding the phylogenetic tree. And this is, do you see the video? This is just a visual impression of the cortical expansion across many different primate species, where brain volume increases and increases and increases. And you see how the folding emerges more and more, how the brain becomes more and more folded.
Please try to wrap up now.
Pardon?
Okay. Please try to wrap up now.
Yeah. Okay. To quantify that, we fit the Gompertz model, and so that we can observe the heterochrony, like the onset of where it starts growing first and how fast. And then we can look at that during development, not during species. So this is a growth trajectory of a rhesus macaque, how it starts growing and how folding gets put in place. And in ferrets, I'm just going to show the video so that you have a visual impression. And so ferrets are even like farther away in terms of speed across species growth.
We will have to stop.
Okay.
We do not have sufficient time, so please come to the conclusion part.
Yes, that's it. So we observe a similar gradient throughout growth and throughout cortical expansion in evolution. And so thank you very much for your attention.
Thank you so much, Katja. It's really a very, very interesting talk. And, it was very fascinating to see how the surface areas and the volumes are changing across evolution. Thank you so much. I guess we have a question for you. The question is from Anastasia. She is asking, how many samples did you use per species? What is the reasonable number of species needed to quantify these relationships?
We used, in most species, we only have, I don't know, one specimen per species, and that's very little, but there's not more available. So for some species, we were able to add 14 specimens, but for most
it's 10 o'clock.
It's 1 or 2 species. And in terms of phylogenetic tree, the estimations get less and less reliable if we have only a little number of species. Also, 34 is the most ever done, but it's in terms of phylogenetic analysis still little. So yeah.
Yeah. There's one more question. I guess we can just put it up here. Means like squeeze it inside the little time that we have. It's from Tilak. He's saying, what would you say to the statement, the degree of folding of auditory cortex increases with complexity of communication? For example, in ferret, it is smooth without folds, but marmoset has 1 to 3 folds per side. Most significant one is the auditory cortex or next to it. So how would sensory deprivation birth at birth affect folding?
Ah, like if you make one more fold, it's like as if you put one more module in place that could give space for one more functionality. So if the anatomy increases and gives you one more option, like adds another module, then you can have more functionality or more diverse functions, and that corresponds with what was observed in cytoarchitectonic studies.
Wow, perfect. Really very amazing talk. And I'm sure you will get more questions from our audience later as well. And now we shift to Daniel.